Okay, I'm going to go ahead and get started. So, again, I guess it's pretty apparent when you guys go and look up your grades, but for anybody that wasn't here, just showing up. The breakdowns apparently are now available for you guys, actually on Blackboard when you go to see them. So a lot of the stats I'm going to put up here, you can see on Blackboard. Again, this was certainly, like I told you guys after last exam, I wanted to make this one a little bit more challenging, at least sort of make you guys have to sort of think a little bit, and certainly the average, average actually didn't change at all, which is kind of interesting. I think more noticeably, because I think there was a few easier questions as well. I tried to put a balance in there, but what you sort of see happen is now we actually have a result, which we really didn't have before, right? It was just kind of like a wave in the last one. Now we really have, I think, a bell curve, in part because, you know, getting above an 80 this time, I think, was probably comparable to getting above a 90 last time. So if you got above an 80 this time, you know, that's, in my mind, similar in terms of how well you did than had you gotten above a 90 last time. So at the same extent, on the other side, it was a little bit easier. And so if you got below like a 30 on this exam, I think that's a little bit more concerning than had you gotten below 30 on the first exam, simply because I think the first few questions were fairly straightforward, and I was hopeful that most people would get that, which most people did, but again. So at this point, again, there's really not too much time left in the semester. Again, we're through, I think, the most difficult component of the course, which is reactions and synthesis. Continue to work on that, specifically if you struggle. There's no reactions, will be a few, but really nothing too challenging at this point in terms of new material for synthesis, and so it's a good opportunity if you are perhaps a little bit behind to really catch up on that stuff during for the final exam. I would certainly, I was just talking to Danielle about, what to cover on Friday, and when she's doing her recitation, and I said, well, at this point you can really cover anything. I would just let the students ask what they'd like you guys to cover, and if you'd like them to continue working on synthesis and stuff like that, I would certainly urge you guys to, you know, say, well, if we've got something you don't really have anything planned, might we be able to go over synthesis and stuff like that, again, because I don't really foresee the remaining material being overly challenging. Again, my hope is it's going to be more culturing in a way. Hopefully you guys can find it enjoyable in terms of that transition to the next sort of course. So again, regrade policy, as always, there'll be a few blatant mistakes if you happen to be one of the lucky few that we just screwed up on. Just talk to your recitation instructor about that, confirm that it is a blatant mistake, and then they can just give it right to me, and I'll make the change, no questions. Well, I'll have to confirm that it was a blatant mistake as well. And then again, any partial credit mistakes, feel free to fill out a regrade request form. Again, not that many people filled out regrade request forms last time, you know, and fine. I mean, I think my whole thing about perhaps getting you guys scared that I'm going to regrade it, right, is that you think that I'm going to automatically go in there and just look for points to take off because I'm angry that you're making me regrade. And that's not really the case, right? The sort of idea is I just want to make sure that if you genuinely feel like you have it right, you know, then you should go ahead and submit it. So I don't want to scare you guys from submitting regrades if you feel like you have something legitimate. And again, I won't, unless it's like staring me in the face, right? Like, oh my God, here's points that are lost, and that kind of happened on the last regrade, where somebody ended up losing points specifically because of the question that they were asking about. But unless I really feel like somebody's just grade grubbing, you know, I'm not going to like go out of my way to try to take points off. So yeah. Just my sense of the exam and the difficulty level of the exam and how well you guys did with it. It shifted a little bit. It shifted. I basically shifted everything a little bit because I figured that, you know, there were some difficult questions, and so especially like, you know, I didn't expect people, you know, I was only expecting a few people to perhaps reach into the 90s, so I really view this as like the exam kind of started at 90, right? So this one, in the same extent, I think, kind of started from 100 in my mind, so that's why I shifted everything back. But on the same extent, from this side, I think there was some easier questions. Yeah.
What are the colors?
When we get into the middle, I don't know. Just green. It's a rainbow, right? So I guess green is kind of A, and I would imagine that sort of the orangish, reddish is kind of the B range, or sorry, the C range, excuse me. Where the sort of purple is borderline D-ish, and the yellow is probably the B, and the sort of light green is sort of B minus, A plus. Sorry, A minus, B plus range. So again, it's just sort of the sense. This is just so you can sort of get a feel for roughly how it is. So that's kind of how I approach it. But again, this isn't how I grade things, you know what I mean? I put the grades out there. I look at them. I look at the finals. When I'm all said and done, I have a set of grades, and I take a look at the final. And so if you guys really respond well in the final, it will help everybody's grades more generally, because what I'll do is I'll grab especially clusters near breakpoints, and I'll say, does this feel like an A? Does this feel like a B? Does this feel like a C? And I sort of will use the final exam as a internal gauge for me. All right. One more thing while I'm on here. So I mentioned this to you guys in the past. I haven't really made anything formally aside from just telling you guys in class. So Monday, I accidentally scheduled a talk at City College. So I'm actually going to be visiting there and giving a seminar. Just friends are up there, and with my 10-year talk upcoming, I just wanted to go there and practice out my talk with them. And I accidentally scheduled it. I wasn't really thinking, but I scheduled it on a Monday. So there's not going to be class on Monday, but what I would like you guys, we sort of discussed a double class or something like that. And I think just to sort of maybe, especially since currently we're kind of culturing, I kind of view this as more culturing, and especially where we are in the class, I just decided this morning that what I'm going to do is I'm going to post the video online for you guys to watch as an alternative, and it's Carolyn Bertozzi, so it's basically click on this link here, and it's going to bring you to this video that she has. And so this is Carolyn Bertozzi. I'm actually really hoping she becomes our Friedman lecturer next year, or one of the upcoming years. I've talked to a few of you guys about her. She's like one of my absolute favorite chemists. And what she works in is an area of, so this is about an hour long. This is about an hour-long video that you guys can watch on your own time. What she is she's a synthetic chemist, so there's overlap there, but she's really one of the probably most, one of the most, I guess to say, biggest name chemical biologists around today. And what that means is it's people that use sort of synthetic chemistry synthesis to sort of probe and understand biological processes. And so one of the things that Carolyn's group, Professor Bertozzi's group, has done is they've developed some really interesting chemical tools to be able to study sort of sugars in a biological system. And she's also a fantastic speaker. And so I think you guys would enjoy that. So there are things within here that could certainly pop up on the final. There's plenty of things in here. She sort of will often give her talks, I think, very often for a general audience, so it won't be necessarily over your guys' heads. Some of it might be a little bit, but I think you'll get something out of it. So I'm going to ask you guys, you're responsible for watching this video, and let me know if you have any questions for it, but I think you guys will enjoy it. And again, there's a link to it just on my website. And again, I think the timing with her talk, the fact that we've been discussing sugars, it just made sense. So, all right. Okay, so today we're going to finish up discussing sugar chemistry. So we sort of introduced it the last time we had class. I guess it was a while ago that we sort of discussed it. Okay, and last class we primarily focused on sort of sugars, the fact that they form these chairs, and so forth, and some of the different aspects behind them. Primarily we talked about sort of six-membered sugars, right? Right, where you have effectively the six-membered ring with an oxygen, again they call it pyranose, and then oftentimes what you'll have, again you may have a series of around them, and again it's simply, I don't know, I'm just writing random OHs in random directions, I have no idea what sugar this is, but again this is the sort of general structure of a sugar, right? You have a series of, you have a sort of a chain of carbons with sort of oxidations, alcohols, or in some cases aldehydes at some of the different positions. Primarily we saw six-membered rings, especially since you guys are going to be, many of you guys are going to go and take biochemistry. I'll just briefly mention that they can also form five-membered rings. These are going to be called pyranoses, right? So instead of six-membered rings, sometimes you'll see five-membered rings. You don't see it nearly as much, but the one sugar that you guys will see it in is sort of ribose, right? And so ribose this is one form of ribose. I'm just going to show it in the pyranose form. And again, you're going to see this in DNA and RNA in terms of the oligonucleotides that we see with it. It's important to recognize that even in cases like ribose, where we'll begin to see sugars, really the sort of pyranose form really only represents about 20% if you just sort of let it stir in solution and sort of got an equilibrium. Really what you're really only going to have is about, especially if I do this, squiggle line there, if I can do either or. This is only really going to account for about 20% of the sort of ribose that you see in say, equilibrium. The remaining component, again, will be the sort of six-membered ring. So, but again, you'll see ribose on occasion, but as you might imagine, right, a six-membered ring is probably going to be a little bit more stable than a five-membered ring. So again, this would be 80% in solution, but what you'll see it in context of again, oligonucleotides, these are instances where obviously there are other factors at play. They'll be capped so that they can't necessarily, you'll have functionalization here so that it can't necessarily undergo that equilibration. Okay, so that's just, this is ribose. You're going to see it next semester. Recognize that it does come from a sugar. And again, it's just the only reason I'm even mentioning this because it's a biochemistry course that you guys are going to take, and to recognize that that is sugar-based. Okay. The other thing last class we started discussing is some of the chemistry associated with these molecules, and I started to rush through it a little bit because we had a lot of time. So I'll just sort of take some time and go through some of the chemistry that we'll see that is associated with this. I'll show later on, actually, some of the challenges associated with sugar chemistry. I think it's kind of one of these interesting things. Synthesis is often considered sort of, one of my biology friends was kind of like, can't you guys synthesize anything? And it's like, no, we can in principle with infinite resources, but I think sugar chemistry, as we'll discuss, is one of these issues, and I'll show you an example of how synthesizing sugars can be very challenging for reasons that you guys might be able to understand, but I'll try to put it in perspective. But at least in terms of the book's chemistry, there are a few different types of reactions that are, again, fairly straightforward based upon what you guys have seen in the past, right? And so if you take, say, for example, cyclic hemiacetal, right, and you treat this to, say, for example, an alcohol in the presence of water, in the presence of, say, for example, an acid. As you guys can imagine, this is a process that you can probably all predict. You can expect that predictive nature under these acidic conditions. You guys can probably all draw a mechanism. depending upon, there's sort of multiple ways that you could draw it. You could either protonate this oxygen, generate the oxonium, and then have the alcohol kick back in. Or the other thing you could do is you could protonate this oxygen, have the aldehyde kick down, and perhaps head that into the aldehyde. But there's multiple different mechanisms that you can imagine. But at the end of the day, again, this is going to be likely driven by the release of water. And so whichever mechanism you prefer, whichever mechanism you draw, what's going to happen in these cases is that we're going to actually make this what we call glycosidic bond. So this is exactly the name of it. We can call it glycosidic bond formation. Okay, and certainly there's other reactions we can do, specifically when we're talking about these sort of anomeric carbons, right? So obviously here I'm not showing a lot of the other alcohols. We'll discuss that a little bit later on in the class. The other thing that you would certainly be able to predict in the course is that if you have, same again, anomeric hemiacetal. If you took this molecule and you treated it with, say, for example, a hydride, whether it's lithium aluminum hydride, sodium borohydride, or the like. Right, if we took this molecule and treated it with sodium borohydride, in its current form, you might not think that it would react. Maybe it might just be protonated, right? But if you sort of recognize that this hemiacetal is in equilibrium with what? The open aldehyde form, right? You can sort of realize that since this is in equilibrium with the aldehyde, right? If you sort of do and you sort of look at the aldehyde, you would all be able to sort of know at this point, given your knowledge of organic chemistry, that this would then reduce. Oh my God, this is kind of horrible. Right? And again, following an aqueous workup, you'll protonate that once you actually reduce it. So simply another reaction. Again, very predictable based upon what you've seen previously, hopefully, right, from the carbonyl chemistry chapter, or although you might be able to come up with alternative mechanisms. Again, in this case, same thing, carbonyl chemistry chapter. Again, both those instances kind of thinking about it in terms of how it could come from the aldehyde form. A few of the other reactions the book discusses. One of them is the Tollens test. Did you really? Did you do it like on a big scale though? You're using it to So
this is the classic Tollens test.
You guys see it?
Where we can test chemically for something called a reducing sugar. So I've got dextrose here. And dextrose is a reducing sugar because it can act as a reducing agent. It itself gets oxidized, which means it loses electrons and push its electrons onto the silver plus ions that are right here. And the silver plus ions gain electrons, they'll become silver zero. And anytime you have a metal becoming a zero charge, it becomes the solid form of that metal.
So this is going to be an oxidation reaction, basically, where the silver is effectively going to give off, it's going to basically take on a few electrons to oxidize.
Solution of silver ions, I'm going to make solid silver plate on here because I have a reducing agent and an oxidizing agent that will pass electrons from the sugar to the silver. Okay, so reducing sugar. And there's many types, and dextrose is one of them. So let's do this reaction. So I'm going to start with my reducing sugar, the reducer, okay, which is the
My undergrad, one of my graduate
Okay.
mentors used to do this in front of the whole class.
Transfer to silver ions. And then I'm going to add a hydroxide. It's going to be happening in the basic environment to balance out of redox reactions. And now I'm going to put a top on, and what I'm going to do is shake, shake, shake. And I'm going to shake this glass wrap. And hopefully, if I make this reaction carefully, all right, we should see some plating occurring. Okay. Takes a few minutes, so I'm going to shake. And hopefully you can see a chemical reaction taking place. And that solid silver
Getting busy.
up here. You see there's a reflection.
This could be bad if somebody had like.
You can see that they're probably making a solid. I'm just shaking to finish this off and spread out.
Okay, we get it. Like kidding.
Also good.
Pretty cool, right?
Silver.
There's the guy. So again, that's the idea. And again, I had one of the guys that I used to TA for when I was a graduate student at college used to do all of these experiments in front of the class. And, I don't know, I'd be a little bit nervous about, again, transporting chemicals down the lab, down the hallways from college. But again, just to sort of explain what was happening there that we saw in the video, again, we're starting specifically from what's happening here is formally an oxidation. Okay, so what the mechanism is, don't worry about that in this case. Oh my God. Okay, so formally what's going to happen here is simply an oxidation, right? So I'm just going to write that for sort of just my little abbreviation for an oxidation process. And the reagent is effectively going to be cationic silver. Okay, so it's going to be species like this, which is cationic silver, which effectively means that it can draw on electrons, it can take on electrons, as you sort of showed there. I believe you said there was also sodium hydroxide in there. But again, the sort of point here being is that the net result, and you're probably never going to use this in a synthesis problem, although now that I've said that, you probably will. But what you're really doing is you're oxidizing this sort of masked aldehyde up to the carboxylic acid. And the reason why this was perhaps useful to people early on was, again, when you had the silver in there, you would get that very quick readout, which I guess is something that you guys do in your labs. Do you do it to test for an aldehyde? Okay, so I guess that would make sense, right? So again, the idea is this is a masked aldehyde, and it becomes very obvious and apparent. Now, in terms of, I don't want to talk about that. The one last thing that we'll see sometimes is, again, we discussed this last, I think we might have begun to talk about this a little bit last week. Again, our molecules have lots of alcohols on them, right? And so throughout molecules, you'll have sort of 1,2 diols throughout these entire systems, right? And if you guys remember, if you take something like a 1,2 diol and you treat it with puritic acid, right? What's going to happen is, anybody remember? Yeah, we're going to get this aldehyde, right? So we're going to cleave that bond. This is sort of the sort of dihydroxylation puritic acid cleavage is kind of like the two-step process, if you will, for like an ozonolysis. So it's analogous to that in terms of synthetic strategies. Now the thing with puritic acid, right, in terms of this cleavage event, is that what needs to happen is that you need to, in terms of the mechanism, you actually need to generate a sort of five-membered ring. So the iodide generates this five-membered ring in this system. Right, the iodide is going to be at a fairly high oxidation state. Think about this. So it's going to be something like this, right? So 1, 2, 3, 4. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 6, 7. Okay, so it's going to be something like this. It's going to be iodide in this sort of oxidation state of 7. And then what's going to happen in this process, you're going to end up dumping electrons into the iodide, and in that process breaking those bonds. But the key here is that you need to get to this cyclic complex in order for this subsequent mechanism to take place. So again, you're going to go from iodide 7 to iodide 5. 7, yeah. Okay. And so in the context of sugar chemistry, what's important about this, and what's sort of relevant about this, is that in order for you to perform this type of event, you need a certain orientation of the molecule. So if we just think about this reaction on cyclic diols, if you have sort of syn diols, right, where they're on the same side of one another, you can form this ring system, right? If you think about the sort of 6-5 ring system, you can generate that. Right? So you can sort of think about this in terms of a 6-5 ring system. But if you imagine this being trans, and this is fine, again, if you went ahead and tried to draw a model of this, it would be fine. But the challenge is that if you guys have a trans diol, and again, you can perhaps, it's not perhaps immediately obvious, right, because I'm drawing it on a two-dimensional board, but if you went ahead and got a model out, what you would sort of notice is that the ability to do the same type of well, I'll show it. So this is the key to this. So it can, I see what you're saying, but the energetics, right? So let's talk about why this is the case, right? So if we have a, say for example, a 6-membered ring, oh yeah. Hold on. This would be the chair. Okay, so I guess technically, but the energetic difference between the two is, hold on a second. Hold on a second. No, Belly just brought up a point that I think is very valid, right? That it should be able to do this. But yet I have written down in my notes that you need it syn. So the question is sort of, why did I write that down in my notebook? I do know that it's much slower, and I do know that, say, for example, trans, especially trans 5 ring systems, for example, you can't do, right? So it's, well, there are examples of it, but it's very rare because it's so high in energy. Certainly a trans 5-6 is lower in energy than the cis 5-6. So if you actually allow things to go to, say, for example, thermodynamic, am I just making stuff up here? Could be. But anyway, my understanding was that you needed these to be syn, but now that Chabeli brought this up, I'm going to say, forget that I said that, because I can't actually legitimately say that now that we're drawing this. So anyway, but the important point is that you do need the two diols, and I'm fairly certain that it's in this reaction. I thought that you need it to be said, and I had that written down. I don't know why I would have written that down if I'm... Okay, so it would have to be locked in. So we'll get back to that. So what Chabelli's saying is that if you look at this guy here, if you think about the chair flip to it, maybe that's what I was thinking, is that you notice that both of these alcohols are equatorial, right? So if you're locked in a situation like that, certainly you wouldn't be able to sort of undergo them. So maybe I just misread that briefly, because you're 100% right. You should be able to. Anyway, let's move on. Why is this important? Why do we care about this process? The reason why we care about this process is it allows us to perhaps degrade sugars and get to smaller components of them that could perhaps be valuable in terms of our sort of understanding the sugars. And so if you, say, for example, want to know if a molecule is specifically in cases where you have, say, for example, methoxy, right? So once you've got that glycosidic bond forming event, so if you have, say, for example, a 5-member ring, maybe this is what I was thinking about in furanosis. Say, for example, if you had a molecule like this, right, and you wanted to degrade it down. If you wanted to have a molecule like this, and you had a certain molecular mass associated with it, and you weren't sure if it was the furanose or, say, for example, the respective hold on. Shoot, because I did this. Sorry, guys. Give me a second. Okay, so if you had this sugar, sorry, I was just on the wrong side. So what we want to do is basically switch that one up with that one. Now we're going to have this reversal. So we're going to have this guy down here. So if this opens, flip it around. Okay, so these would be two possible isomers of one another, right? And if you're, say, for example, trying to figure out which one is which, one of the things that you could imagine doing is trying to break this down through periodic acid cleavage. And what will happen is you'll be left with smaller fragments. You'll liberate things such as carbon monoxide and carbon dioxide and certain formates. And so what you would sort of do in this case is when you look at a molecule like this and you treat this with, say, for example, pyruvic acid, you see the 1,2 diol there, right, which will undergo cleavage, right? And so if you were, say, for example, use mass spec in this molecule, following HiO4, you'd cleave to that molecule there, but you wouldn't actually change the mass other than what you would do from the oxidation. So it would be a very subtle mass loss. You would just lose effectively one equivalent of hydrogen, right? So the mass would be different. But if you were to do the same thing on a system like this over here, you have a couple different diols, right? So you have the diol that you see here. You have the syn, so you have the diol here. But at each of these positions, what you could in fact do is begin to oxidize them. Okay, and so if we look at this molecule, if we think about the first oxidation here, in this case, what's going to happen, right, is that we're going to get no, because this is the CH2 here. Yeah. I just almost accidentally drew that. Okay, so you would do the first oxidation, but then again, in an aqueous solution where you have oxidants and stuff like that, what can happen, and again, we see this, say, for example, in oxidations with Jones reagent, right? If you have an aldehyde, you can oxidize up to the carboxylic acid, because what's formally going to happen is these can exist in equilibrium with the hydrate, right? And once those are in equilibrium with the hydrate, right, then what ends up happening is you can have a situation where now, in fact, you can break that bond through periodic acid cleavage as well. In this case, what's going to happen is you're going to generate a smaller mass compound, and again, if you add the hydroxyl in and do that cleavage, what's going to happen is you're going to generate what? Formic acid, right? So what will happen here is you'll have formic acid. Okay. So again, similar isomers of one another. If you're trying to figure out which one it is, apparently periodic acid cleavage is actually used to try to sort of understand these a little bit more. I want to talk about more interesting stuff. Something that I think is very valuable, so I'm going to move on from the book chemistry and talk about things I'd rather talk about. And one of the things I think is really important to recognize, just sort of more historically, is what an important role sugars played, specifically in our understandings of things like stereochemistry. And so we discussed this a little bit more in depth. No. It's fine. Ignore that I said that. Because it should be. I don't want to confuse matters. I don't even really care that much about this. What I'm going to start talking about a little bit more in depth is things that I think are, the whole point of this is, I think, to culture you. So the idea here is, again, that oxidations could be used to effectively decide between the two. And so just go nuts. 1,2 diols, we'll just leave it at that. And because, I don't know why I had that written down. Anyway, as we sort of stated earlier, sugars, I think one of their early and really important applications was in sort of understanding stereochemistry. And I think one of the things that your book doesn't have, but I think is really important to mention, the Kilani-Fischer synthesis. And it was a method developed by Fischer back in the day when they were trying to actually understand the different stereochemistry behind these different molecules. And this goes back to the discussion that I think we had a little over a week ago about, well, why is this D and why is this L? And part of the reason was that when we were taking these sort of aldoses, sort of smaller components that happen to have one rotational barrier, they were given names, and all the molecules that were derived for them simply were given the same D or L annotation. So if you started with a molecule this, you can begin to actually branch and create sort of longer carbon chains while sort of preserving sort of sugar structure to it, right? Because as we all know, aldehydes are electrophilic in nature, right? And one of the ways that we know that we can in fact make carbon-carbon bonds, right, is something that we've seen time and time again, and that's going to be cyanide, right? So if you take an aldehyde of this sort and treat it with cyanide, what's going to happen in this reaction, right? You're going to add it to that aldehyde. Now when you do this, right, what's going to happen is you're going to get two possible stereoisomers. Right, it's going to be non-selective in nature, but it's not going to touch the bottom stereo center. Okay, so this guy down here will effectively remain unchanged, right, from the starting material, but you're going to add into that aldehyde. You can add into one of two ways, right? And so what's going to happen is you start with a single enantiomer, and you're going to get two diastereomers through subsequent reaction. Now, if you look at the cyano group, right, what you'll sort of notice in it is that it's, you guys can probably predict how you would then subsequently take a cyano group and then convert it to an aldehyde, right? If I gave that as a synthesis question, perhaps you guys could predict methods. The classical way that people actually did this, one of the initial ways that they did this was actually to take these molecules and treat them with heat and water to effectively hydrolyze the cyano groups, right? So you can actually make the carboxylic acids. Once you're at the carboxylic acids, those can, if they get long enough, actually form the lactones and so forth. But again, once you're at that carboxylic acid, then I guess more classically nowadays we could say, for example, treat it with diol or something like that to get to the aldehyde. But at the time, what they ended up doing was they treated it with sodium and mercury. Okay, and in this case, what will end up happening is that you'll get to new aldehydes, right? New carbohydrates. And if you continue this process, so if you did this and you went ahead and isolated these molecules, if you made these two molecules, went ahead and separated them, isolated them, and then compared them to, say, for example, natural sugars. What were the natural sugars that had similar properties to them? Which ones had similar melting points? You did melting point suppression type strategies and so forth. And then keep going, right? And then you can take this one and you can sort of drive it further. And it was really, this is some of the earliest sort of total synthesis work, right? Target-oriented total synthesis was really started with this type of chemistry, again, driven by the sort of ability to relate these sugars to one another, figure out how they might be structurally similar to one another, and so forth. Yeah.
Sodium and mercury would be carboxylic acids to aldehyde?
Apparently, well, how do you know to make aldehydes? Stick with what you know. I'm just showing you this was like one of the classic examples. Yeah. But what I have specifically written down here actually comes from the esters, so it might be something specific to esters. The classic way that they do it now, this is sort of an improved, because the problem with this one was that they used to get like very, very low yielding processes. And you can imagine as you're making a sugar and then you're trying to make more and more and more, it's 3 steps to get to this, and then it's 3 steps to get to the next one, and then it's 3 steps to get to the next one. And once you start getting to sugars that we typically think of as like 6 carbons, you're talking about a very low yielding reaction. And so the higher the method, if you want to actually use this type of approach from a cyano in synthesis, you can do LAH reductions of cyanos and then they'll hydrolyze an amine to the aldehydes. Yeah, so cyanos can be, they're really good in terms of diol reductions and so forth. So you could just diol reduce it and hydrolyze from the cyano. I'm not sure they actually use diol. Traditionally, I think they might actually use certain palladium, I think they might use certain metal catalysts. That might be the industrial process through which they do this nowadays to generate sort of unnatural sugars and stuff like that. But I don't really see any use for you having to do this, which is kind of what I'm saying. There's no need to learn this new reaction just to have a fancy method. And specifically, what I'm seeing in the book is you can imagine it wouldn't really work here, but once you get to larger sugars, it could form lactones, right? It can cyclize. And certainly we know that the reactivity of lactones versus, say, for example, esters and so forth are different. And so I'm not necessarily sure that this would work for a carboxylic acid. But the point being, more generally, the idea here that I'm trying to convey is that you can extend, right, from aldoses, sort of one carbon at a time, to generate two new stereoisomers. And they use this to sort of, back in the day, when they were trying to understand sugars and understand the structures behind them, they used this approach, comparing to the natural sources, to sort of figure out what the relationships were behind a lot of the sugars and really gain insight, specifically stereochemistry, right? Because this is one of the early examples where people were really trying to understand stereochemistry and the relationship between different stereoisomers and so forth. So historically very important Nobel Prize work. So extremely, extremely important. Okay. Now, this is sort of classical sugar chemistry. And again, even though this synthesis of sugars has been something that's been of interest for over a century now, I would argue that sugar chemistry remains probably one of the biggest challenges, specifically when we're talking about making, say, for example, oligosaccharides, right? So what I mean by oligosaccharides is sugars that are effectively combined together through a series of glycosidic bond-forming events. And so one example of what I'm specifically referring to is to sort of put into perspective how difficult sugar chemistry can be. What's that? Yeah, so it's more than one. Yeah. So well, polysaccharides, oligosaccharides suggest certain. When I think of polysaccharides, I actually don't know the inherent definition between the two, so I don't know the semantics behind it. But when I think of polysaccharides, I think of things like cytokine, right, which is like the shellfish. Like what shellfish stuff is made of, and it's usually a repeating unit, right, of the same sugar. When I think of oligosaccharides, they can still be longer, but what we're talking about is some variability, so not necessarily always the same sugar. Because when we talk about polymers, we talk about the same sequence being repeated over and over and over again. Oligosaccharides just means multiple, I don't know if it's short, but multiple sugars. I'm just going to put up a trisaccharide, just kind of an oligosaccharide, and you'll see it's not overly. Okay, so this, what I'm drawing here is a sugar, and I'll explain why this is relevant to me. So when I was in graduate school, I was working on a project that was really cool, I thought, people think, that involved here's a trisaccharide. So basically 3 sugars that are sort of bound together. So when I was in graduate school, let me take even a further step back. This will become clear when you guys watch the Bertozzi video that I posted for you guys. But one of the reasons why sugars are really important biologically is that cells generally will often have, you guys sort of perhaps know this, but cells will often have sugars displayed on their surface, covalently linked to the proteins on the surfaces of cells. What's that? Yeah, well, What's that? Yeah, they're everywhere. So the thing is that within, say, for example, immunity, if anybody's interested in like immunology and so forth, sugars can really sort of serve as an indicator for our immune system. And what I mean by that is, you guys have heard of, say, for example, AB blood type, right? I'm not going to have time to go through this, but if you guys would be interested in reading into it, or you want me to send you some information on this, it turns out that the AB blood types that we talk, the A type blood type, B blood type, AB blood type, what that effectively comes down to is the sugars that you might have on your cells versus the sugars that you might have on your cells, and whether they would be the same types of sugars. So there's really two sugars in particular. Some people have both of these sugars in them. Some people have neither of them. Some people have one or the other. And depending upon which group you are in, if you get somebody else's blood, there's the possibility that your body might not be prepared for those new sugars on the cells. And the reason behind that is, right, that our bodies, whatever is natural to us, we basically deal with, but we have obviously ways to fight off pathogens and foreign things. And if we sort of recognize something as foreign, then our body's going to want to clear it out. And so if that's not native to you, then your body might be primed to have a response, which could lead to, say, for example, anaphylactic shock if you have a blood transfusion and suddenly you're pumping in foreign stuff into that person. In my research group, sorry, not my research group, during my postdoc, in the research group that I was in during my postdoc at Yale, one of the things that we were interested in is how might we leverage some of the antibodies that are already present in the immune system to, say, for example, redirect immune responses towards things that we would want, right? So we're talking about the fact that you have antibodies in your bloodstream already that are perhaps primed to go after something foreign, right? And one of these sugars that our body is often primed for is, well, not this full molecule, but this part over here, which is called alpha-gal. And alpha-gal is a sugar that's found on basically everything, right? Like like every organism in the world besides humans have this sugar associated with them, including the bacteria in our guts. And so we are constantly being exposed to this, what we call antigen, but our body still recognizes it as foreign. So we always have these antibodies that are like hanging around in our body, which anytime they recognize this, they go after it and they try to basically take it out, right? And this has been a big problem in, say, for example, organ transplants. If you're going from getting a pig organ to put into a human, right, the pig will have these things and people will start to reject the organ and things like that. So people have known about this for a while. What's that? Humans don't have this, right? It's like us and like third world monkeys or something like that, right? Like it's yeah, basically everybody but us and like a couple monkeys. But anyway, the question that was sort of that my PI had, and other people had this in the past, including Laura Kiesling from Wisconsin, somebody that we had talked about, and that's actually where this paper had come from. People were interested in whether we could make molecules that would have something like this that would target the antibodies in your bloodstream, and then have something over here that would, say, for example, bind to a cancer cell, or that would bind to some bacterial cell, or, say, for example, an HIV or something like that, right? And effectively elicit immune response to target that cell. So anyway, so that's just the background. But when you look at a sugar like this, right, you can't buy this. You actually have to make this. And when I was a postdoc, I had thankfully gotten my first paper out and was on my way to getting a job here. But we were kind of curious about, we hadn't used this antigen, we were using something else, a little bit simpler one, but we were kind of curious about studying this. And so I ended up having to make this molecule, and I've been doing synthesis for 7, 8, 9 years at that point. And I was tasked with synthesizing this molecule. Now, formally, all that I had to do, right, with this molecule, oops, wrong paper, is starting from 2 sugars. So here's 1 sugar. And it turns out that this sugar over here is actually lactose. So we could buy these 2. That was okay. But we needed to end up linking these 2. Okay, so this is this sugar, this is this sugar. I could buy tons of both of these. The only thing that I had to do, right, was make a single bond, right? And that's from this, right? That's from this carbon here. We need to make a glycosidic bond forming event between there and basically this. Okay? Simple, right? It took like 10 steps, right? It's 1 bond, 10 steps, because again, like you're saying, yeah, it just said that we are effectively asking for this 1 alcohol. Even if we can make this molecule, that would be pretty easy, right? We could protect all of these, and that's a glycosidic bond. So making this into electrophile is pretty easy. The question becomes, how do we figure out exactly how to make this alcohol out of all of the rest of these react? But people have actually figured out a way, and so I thought it would be kind of interesting to show you guys. I'm not going to go into too much depth, but I'll at least show you the strategy, which could have actually resulted from some of the confusion that I had that I was talking about earlier. Maybe this was on my mind, because you'll see what I'm talking about. And so herein lies the issue. And so what we had to do as a strategy was basically take this entire molecule and protect every single one of these hydroxyls except for this one. And the way in which we do this, so effectively this is what we wanted to make. A benzyl substituted version of this. And again, this isn't us, this is actually, this is protecting something else. This was a strategy that had been used. In fact, we were following a prep by Laura Kiesling, who I mentioned earlier, but it was really this molecule that we wanted to make. So what we wanted to do is protect all of these different hydroxyls and leave this one wide open. Well, it turns out that there's a kind of interesting trick that synthetic chemists can use, and it does have to do with that sort of cis versus trans 5-5 ring system. Okay, and if you look at the molecule that I drew over here, every single one of these hydroxyls, so if you can imagine that they're the free hydroxyl, every single one of them, except for one set, is actually trans to one another. Okay? There's only 2 hydroxyls in this whole series that are cis. And where are those? Right here. So these are the only 2 cis diols. All the rest are trans. And as it turns out, if you take these molecules, if you take that free acid. I won't draw the other part, I'll just show the relevant part here, and you guys can hopefully forgive me. Again, this is, and everything else has, again, this is trans, this is cis. And it turns out if you take this molecule and you treat it to dibutyl tin oxide, okay, in the presence of like molecular cis, and you heat it really, really hot, you can think about this as being a carbonyl. Right? And you can think about a condensation reaction, at least mechanistically we can, but at least in terms of something that you guys are sort of used to thinking about, you can think about this as being like acetone in a way. And if acetone was going to condense on, say, for example, a few of these alcohols, where it's actually going to condense is going to be here. Okay, you can actually form that tin bicycle. And again, this is the most stable conformation in the ring. And then what you can do once you make this is now you've effectively narrowed down the choices of nucleophilic. So it's not even, it's still not that simple, because at this point we've narrowed it down to two sort of oxygens, right? And based upon the tin bond, what do we know about tin carbon bonds, for example? We think about them, at least in terms of transition metal catalyzed chemistry, as being somewhat nucleophilic, right? And so it turns out that now by coordinating these to tin, these oxygens have a little bit more nucleophilic behavior to them. And so we can, in principle, react them with a nucleophile. And the nucleophile that we're going to react them with is actually going to be allobromide. And axial, as you might imagine, is actually going to be less sterically accessible than, say, for example, the equatorial. So it turns out that this equatorial oxygen is more accessible, and again, we can actually react it with it. And so what we did was we took this, treated it with allobromide. Okay, why allobromide? Okay, and again, we haven't really learned this, but allyl groups are another class of protecting groups. So what we can then do, once this is protected as an allyl group, is we can benzylate everything else and then deallylate it. And this is the problem with sugar chemistry, right? That's a pretty fancy way to select, but it involves all this protection, deprotection, protection, deprotection. Ways to simply take a molecule like this, right, and just have it make a bond with this guy here, using that oxygen, at that specific oxygen, is where synthetic chemists were just not really good. Yeah?
So what was the deal? Would it go two ways?
No, it's more open here. It's more accessible here. Yeah. So if there was some of this, I didn't notice it. Yeah?
Right up to that, the oxygen attached to the, yes. Next, yeah.
6-member ring.
Yeah.
Yeah, so great question. And I don't have a great answer to that. It may just have to do with maybe there's some type of an entropic penalty that's paid, right? Because that's got complete freedom, right? And maybe once you bind it to it, now you've sort of locked it. And things like to have some movement where everything else is sort of already locked in that position. So that might be the reason. But it's a good question. It's a fantastic question. Well, because we want to try to isolate it. So it's a thermodynamic product, and then it would be over here, right? So it might just want to flip over to the other side, just because restricting that might just have some energetic problems. Yeah. Well, that's where I was going to go again, because you guys are going to go and take a biochemistry courses, and this is one of the really important sort of recognitions. We're all finishing synthetic organic chemistry right now, And the question was just brought up, well, what about enzymes? Clearly this bond is made, right, in nature. And again, one of this sort of thing that you guys will hear, the video, and you'll watch Carolyn Bertozzi's video, she'll talk a little bit about enzymes that actually make these glycosidic bond forming events. And part of her research is sort of how to study that. But yeah, this is where enzymes, synthetic chemistry is great, I think, right? But yeah, but it's great in that we often develop methods that can be used to do a lot of different things. Whereas enzymes are fine-tuned to do a specific task. Oftentimes a specific reaction on a specific protein or a specific sugar. And so exactly, they're fine-tuned towards selectivity. Whereas oftentimes synthetic chemistry developments are done with having in mind, I want to be able to do this in lots of different scenarios. And so in a scenario like sugars, this is, I think, where synthetic chemists have a sort of a long way to go. And there are people that are trying to sort of solve these tasks and sort of so forth. So again, this is sort of an example, and again, just some of the trickery that needs to be done to overcome sort of synthesis. Okay, any questions? All right, so I don't want to really get started on anything new because we only have about 5 minutes left or so. So I'm going to let you guys go a little early. So again, for those of you that showed up late, I'm not going to be here Monday. I've got a video on my website. Go watch it. You're responsible for it. It's a good video, so you'll enjoy it, I think. And I'll see you guys on Wednesday. Yeah.
When are the off hours?
Now.